Okay, folks, this is part one of our biotechnology PowerPoint video, techniques for producing and analyzing DNA. This is not completely comprehensive, but it gives us a lot of the bits and pieces that we need to understand things that we'll be talking about in class. The first half of this presentation, which is what you're watching now, deals with some of the biotechnological tools, so some of the things that we need to use to be able to manipulate DNA in the way that we wish to. The second half of this PowerPoint presentation video is going to be about how we're going to use some of those tools to carry out particular techniques that we use for maybe mapping DNA or for sequencing DNA or for copying DNA and so on. So some of the many things that we can do to manipulate DNA in the lab. Okay, the first thing that we're going to talk about are restriction enzymes or restriction endonucleases. And they're very, very useful enzymes. These enzymes are unique in the way that they are able to cut pieces of DNA. So we can think of them as being molecular scissors, and like any enzyme that we've learned about, they are absolutely specific. They cut DNA, but they will only cut if they have a specific pattern that they're able to hold on to. So if there's not the right sequence of A's and T's and C's and G's, the restriction endonuclease can't bind and the cut will not be made. This particular pattern that each enzyme requires is called a recognition site, usually is between 4 and 8 base pairs, and the sequences are palindromes, and I'm sure you know what those are, and we'll be looking at some, so you'll see a few examples. The one that I'm showing you here, the EcoR1 cut, if you, again, line them up, so C is over G, A is over T, A over T, T over A, T over A, and C over G, you will see that they read the same front to back, GAA-TTC on the top, and from the 5' at the bottom, GAA-TTC on the bottom. And of course, they're base paired. So here's a better look at it, the graphic that we have here, 5' on the top to 3' from left to right, and 3' to 5' on the bottom. And we can see C paired with G, A with T, A with T, T with A, T with A, and C with G. And if you read in the 5' direction on the top, it's GAA-TTC. If you read from the 5' direction on the bottom, GAA-TTC. So they are palindromic. And then we see a blue blob on our graphic that's representing the EcoR1 restriction endonuclease, and you can see that it's performed a cut in that sequence. So between the G and A on the top strand and the G and A on the bottom strand, cutting the phosphodiester linkage on the backbone and allowing the hydrogen bonds to break between the remaining base pairs. So the 2 pairs of DNA, or the 2 strands that we've now created of DNA, have these 4 base pairs that are not bonded to another strand. So they're sort of hanging out there, these little pieces we call sticky ends. Okay, so again, as I mentioned on the previous slide, they are able to hydrolyze, add water back into, and therefore break phosphodiester linkages between the nucleotides, and this disrupts the hydrogen bonding that's occurred between the base pairs. If we have the staggered cut like we saw with EcoR1, then we have sticky ends that are left. And sometimes certain enzymes will cut blunt ends where each piece is fully base paired. So if you have a look at the cut marks here, the first 2 enzymes create blunt ends because they just cut straight through, where the last 3 enzymes cut into specific base pairs, disrupt the hydrogen bonds, and leave sticky ends. So different enzymes are able to do different cuts, and as a geneticist or a molecular geneticist, you'd be able to make cuts in the DNA precisely where you want to by choosing the right restriction enzyme. All right. So again, it gives us a lot of options. Because it's so specific, we can predict where particular DNA fragments that we're investigating are going to be cut, and what's going to happen, what sort of sticky ends they might have, and what we might be able to stick in there if we wanted to add some pieces of DNA into a piece that we already have. Okay, again, if we do have the sticky ends, we get something that we can very specifically bind into or have other pieces of DNA that we're working with connected to or anneal to the pieces of sticky ends that we're creating here. Where blunt ends, we can pair up with all kinds of different things, but we may not be able to be as specific with the blunt end combination. So it depends on the intent of the geneticist as to whether he or she will be using a blunt end type endonuclease or one that's going to give us sticky ends. All right, so what the heck are these things anyway, and where did they come from? Well, they are naturally occurring in bacteria. So as we were discovering the ins and outs of bacteria, we realized that they had this really, I've called it crude here, but pretty basic immune response. And in grade 11 biology, you learned about the idea of bacteriophage viruses that can attack bacteria and inject their own DNA into the bacterial cell and take over the mechanism of the bacterial cell, and instead of doing what the bacteria needs it to, or allowing the DNA to direct what the bacteria needs it to do, they take over and start making virus particles, both protein coats and more of their either DNA or RNA. So the bacteria in response evolved to have these restriction endonucleases. Various ones can be found in various bacteria that are able to cut pieces of DNA into fragments. And the deal is then if we can cut that bacteriophage DNA into lots of different little pieces, then it won't be able to take over the bacterial DNA and direct its own production. So it's the bacteria's way of fighting against invading DNA. So this diagram might look familiar to you from last year, where you have the bacteriophage. It looks a little bit like a NASA landing craft. It lands on the outside of the bacterial wall, which is represented here by this blue sort of straight line. The virus is able to inject its DNA right into the bacteria, and that DNA is going to try to take over the bacterial machinery and use it for its own purposes. Each one of our endonucleases are named for the bacteria from which they've been extracted. So EcoR1 comes from E. coli bacteria, a particular R strain of bacteria, and it's EcoR1 because it was the first restriction enzyme found in this particular strain of E. coli. So literally named sort of as they're discovered. All right, I want you to think about that for a minute though. If bacteria can produce this enzyme that's going to try to cut up pieces of DNA, how do they keep themselves from being victim or falling victim to this? Because the viral DNA is no different really from the bacterial DNA in the sense that, we've got the A's and T's and C's and G's, and nucleotides are the same. So how does the bacteria protect itself from its own enzyme, protect itself from having its own DNA digested? What it does is that on specific areas of the DNA where we have the particular recognition sites, the bacteria basically place methyl groups on the DNA in those locations, which prevent the endonucleases from binding to the DNA and cutting it open. So we have methylase enzymes that can produce methyl groups or place these methyl groups onto the DNA and prevent them from being cleaved or cut apart. So if you have a look at the diagram on the side here, we're seeing that onto one of the nitrogenous bases, we have a CH3 group that's been added. This is going to change the shape change the reactant, if you will, that the enzyme is looking for, change the shape of the reactant. So the enzyme is no longer the right shape to cleave that particular piece of DNA. So quite clever on the part of the bacteria. So they methylate their DNA and prevent the restriction enzymes from cutting the DNA. And we use this tool, the methylases, to produce methyl groups to protect the DNA that we're working with. So if there's particular spots on the DNA that we wish to have cleaved by a recognition or by a restriction enzyme, we can protect other regions in this genome by methylating it. So we can selectively methylate what we require to protect, or if there's something that we wish to protect that we're working with, and try and make sure that the restriction enzyme can't cleave it. All right, this is a familiar friend for us, DNA ligase. We spoke about this when we were doing replication. You remember we use it in our own cells to fuse the Okazaki pieces together. DNA ligase is able to fuse or refuse the phosphodiester linkages in the backbone of the DNA strand. And of course, as soon as we have DNA with matching base pairs close to each other, the hydrogen bonds will sort of reform, the DNA will be annealed, and then DNA ligase can repair the area, repair those phosphodiester linkages. So again, we're looking at the annealing, sort of like magnetically attracted to each other, not magnetic, of course, but the hydrogen bonding, attracting the appropriate base pairs together, and then the DNA ligase can be used to fuse. So if we were trying to introduce a new gene into a piece of already existing DNA, we could cut it with restriction enzymes, as long as it's not methylated. So as long as we haven't protected that area, cut it with restriction enzymes. We could take another piece of DNA and cut it with the same restriction enzyme, so it has the same sticky end pieces, and we could incorporate that DNA into the piece that we cut, and then we could fuse the pieces together using DNA ligase. So think of DNA ligase being like our genetic glue that we can use to stick DNA pieces together that we wish to stick together. Often the ligase that's used to do this is actually a T4 phage, so a bacteriophage or a virus DNA ligase, rather than our own DNA ligase, and it's used pretty consistently in most genetics labs to reform the phosphodiester linkage in ongoing procedures. Right, another one of our tools, and one that we either have already used at UOIT or will be using at UOIT, is gel electrophoresis. Gel electrophoresis is a material that we can use to separate samples of DNA into maybe more manageable bits based on the molecular weight of the fragments that we're looking at. So we can use several kinds of gel, agarose or polyacrylamide. We're going to be making agarose gel in the lab at UOIT. It's kind of like Jello powder. You'll be adding some electrolyte solution, which is pH balanced, and creating sort of literally like a Jello that we're going to harden into something, again, that's very much like a Jello. And we're going to pour the liquid Jello into this little plate that's going to give us wells at one end. So it's almost like a comb that we put into the setup. It's a little hard to see in the diagram that's here, but at one end you would have, when you were making up the gel slab that you see right in the center here, you would have little comb, and then when the Jello is hard, you'd pull a little comb out, and then you have all these little punctures at one end. And what we can do is, in those little punctures, we can add whatever material it is that we're trying to separate, in this case DNA, that maybe we fragmented for some reason. Maybe we're trying to map and see how big the pieces are, see where the restriction sites are on a particular piece of DNA so that we can protect them or not protect them. Again, there's thousands of different purposes we may have for doing this, one of which would be to create a DNA fingerprint, which we can use for forensics work or for determining parentage and so on. So this gel we can create, make the wells, we can put things into the wells, DNA samples into those wells, and then we can run them using an electric current. And the current works really well in this case. because of the charge that we find on DNA. So DNA is negatively charged. If we place a negative charge at one end of the gel and a positive charge at the other end of the gel, of course DNA is going to migrate away from the negative charge. So remember that like charges repel, and toward the positive charge, which would be attractive for the negatively charged DNA. So it actually migrates up the gel. The rate at which it's going to go along the gel is, as it says here, inversely proportional to the logarithmic size of the fragment, meaning in essence that our shorter fragments are going to travel faster. And again, if you think about gel or jello, it's a really big matrix with all kinds of fibrous, interwoven, proteinaceous strands. And the DNA molecule has to work pretty hard to get through it. If you're really big and long, it's going to be bulky and hard to get through. If you're short, you're going to be a lot faster getting to the other end. This is a photograph of one of these gel electrophoresis prints that we made at UOIT last year maybe, or the year before. And each one of these little dashes represent a particular strand of DNA that was run, and actually each sort of strip that we see here is a different sample. So in the case of the gel that we're seeing here, we've got a very long set of bands, a lot of bands on the very left hand, not quite as far left as we could have gone, but the second one in to the left here, we can see a long band. That was a molecular weight marker, so we could tell how far our strands were migrating. The third band over from the left is showed a single strand, and that was basically when the material we were working with was given one cut. So we're seeing something that is the full length there. And in slot number 4, we're seeing it cut with one particular kind of restriction enzyme, and we see that a different banding pattern is formed. There's in fact 3 different bands here. And in the next one over, we're seeing that there are 4 different bands here. And then there's a part that they missed on this section. Anyway, we can see that they're sort of done in sets of 3 with single bands, although some of it didn't work out so well. But this actually tells us something. So by the end of our lab, we should be able to understand what we're seeing on a strip like this in terms of size of strands or size of pieces in a DNA sample. All right, now what we were seeing in that photo was not what it looks like when it comes out of the chamber. In fact, the gel itself, or the DNA that's on it, we need to stain in some way to make the DNA visible. So we can't actually see it. We understand that DNA is soluble in water, and we're not going to see it. We're not precipitating it out here with alcohol like we did in our extraction. So we need to be able to see it. One of the stains that's commonly used, a very effective DNA stand, is called lithium bromide, and as soon as you put it under UV light, it will fluoresce, which makes it nice and obvious to us, so we can see it very well. The problem with lithium bromide, and maybe you can see this from the diagram we have here, is that it sort of squeezes in between in pieces or in areas of the DNA where things are not supposed to squeeze. And basically what will happen is that once we use it to stain, that DNA is completely ineffectual. It cannot be used to do what it should be doing. So it mutates the DNA. And because of this, lithium bromide is actually a carcinogen. So we won't be using lithium bromide, but it is a very common DNA stain for this sort of work. So once it's stained and fluoresced, we can have a look at it, do some measuring, and figure out how big our pieces might be, or compare the fingerprint that we've created with some other, a victim to a perpetrator, or whatever it might be that we need to compare the DNA for, use the DNA for. So it's a very effective tool to measure DNA in lots and lots of different ways. All right, so plasmids are something that you are familiar with, and we're going to talk about them as a tool because we use plasmids all the time in our molecular genetics, because we can use that plasmid. We can engineer a plasmid. We can cut a plasmid where we want to cut it. We can incorporate genes into a plasmid. We can force bacteria to take the or yeast to take the plasmid back up again into their cells, and then they can produce a particular protein for us. So basically, we can force bacteria or yeast, not quite as common, but bacteria, to become little factories for our products. So plasmids are little pieces of DNA. They often contain information that's not vital to the day-to-day workings of the bacteria, but in fact give them added bonus. It's like a I don't know, chocolate brownie at the end of the meal or something. Oftentimes plasmids will contain antibiotic resistant genes. So if you get if the bacteria has a plasmid with an antibiotic resistant gene and you take the antibiotic it's resistant for, then that bacteria does not die. So clearly it's an advantage to the bacteria. What we can do is we can incorporate other genes in there, say like human growth hormone or insulin, and instead of being required to harvest those materials from either other humans, which we really don't do, or in the case of insulin, we used to give diabetics pig insulin. So much better to have human insulin to inject, but people are not going to line up to donate insulin. So we need to have some way of creating it, and this is it. We can take an insulin gene, we can incorporate it into a plasmid, we can pop that back into a bacteria, and I'm making this sound a lot easier than it is, but we can do that, and then we can have a whole set of bacteria, which we know reproduce really rapidly, who can make insulin as a product, and we can literally tap that off and use it to supply people who need it. All right, so as we know, bacteria do themselves have a circular chromosome, and it says here approximately 4 million base pairs, and we can see that labeled on our diagram here. That is the regular genome that they would use to be able to do all the things bacteria need to do. And not all, but some bacteria have this extra plasmid, and the plasmids are much smaller, and like I said, they don't contain genes that are necessary just for the day-to-day living of the bacteria themselves. And that circular DNA can sometimes enter the cell or sometimes exit the cell in a couple of different ways. And those plasmids contain useful pieces of information that the bacteria can use to help in special circumstances. The plasmids themselves are independent of the chromosomal DNA, so they don't replicate necessarily at the same time that the chromosomal DNA will replicate. The plasmids themselves can be replicated up to a particular copy number inside the bacteria, so there can be more than one. Plasmids, as I mentioned, often carry the antibiotic resistance to a particular antibiotic, which is very useful for a bacteria. In some cases, they also contain genes that may allow bacteria to break down heavy metals or things like crude oil. There's lots of bacteria that live in the Gulf of Mexico, naturally occurring bacteria that can break down the crude oil from the leaks in the bottom of the Gulf of Mexico and can use them as a food supply. So all kinds of different things that can be found on that plasmid DNA that can be useful to the bacteria. Here is a look at an engineered plasmid. This plasmid was engineered by people in a lab, and basically we can see there's a couple of different things that we've added in here. It's got in the green arrow, we've got an ampicillin resistant gene. In the red arrow is representing a gene that causes this plasmid to cause a bacteria to fluoresce. So we can tell that bacteria is present just by causing it to fluoresce. We know that it's acquired this particular plasmid. It's a way for us to say, oh, look, it found its way inside that bacteria. It has what we want. Excellent. Let's go ahead and grow some more of those. And then in this case, there's a promoter region, which we would need, of course, to make sure that those genes could be transcribed. And some other gene of interest here for this particular experiment for whatever this pGLO was engineered to do. So some kind of regulatory protein for some function they were investigating. But again, we can we can engineer all kinds of different things into plasmids and then fire them into a bacteria and get the bacteria to do what we want them to do with it. Okay, here we see a bacterial cell with 4 different plasmids and its own circular chromosome in the center. As I mentioned before, plasmids have a particular copy number, which means that for each plasmid, there's a particular number that can comfortably exist in the same cell together. And the higher the copy number, the higher the number of plasmids you're going to find in a bacterial cell. And we, as I mentioned, in the biotech industry rely very heavily on genetically engineered plasmids. These artificial plasmids, which I sort of showed you in the last slide, have been engineered with a whole lot of different things in mind. One thing that they would have that I didn't really mention would be a unique area called a multiple cloning site. And this multiple cloning site would take a whole lot of bases that in combination could be cut by particular restriction enzymes. So they would sort of overlap this area with all kinds of the palindromes that the restriction enzymes can cut through. And that would be the area that's engineered specifically so that we can add new genes into that plasmid area. So we make it easy for us to add in whatever we might want, an insulin gene, a human growth hormone gene, whatever it might be. Okay, there is a little video there. I will post it, the idea of transformation. I'll post it along with this video on the on our D2L, so you can watch it if you want to. You could also watch it. I've posted the plain PowerPoint on the St. Mary website, so you could link it through there as well. So I mentioned earlier that bacteria can often take in DNA or can take in DNA from their environment. Some bacteria are more easily take in the DNA, and if they are a bacteria that, relatively speaking, takes the DNA from other critters up fairly easily, they're called a competent cell. And the way that they, or the process by which they take up the foreign DNA is called transformation. And we can, in the lab, chemically induce competence in a cell, even if it was reluctant by nature to take up foreign DNA. So this diagram on the side here is showing us transformation with either DNA fragments, so just pieces of DNA, or with a plasmid. And again, the idea is that the bacterial cell is, in either case, able to take the information in and either incorporate it into its own DNA if it's a successful transformation, and break it into pieces using a restriction enzyme if it's not. And same with the plasmid, not able to incorporate into its irregular chromosome, but able to take the plasmid up and then be able to reproduce that or replicate that plasmid in the bacterial cell. Right, so the way that we do this in the lab is through a series of steps. First, the bacterial cells are basically chemically and physically frozen. Calcium chloride is going to have an impact on the proteins in the membrane, and the colder temperature means the fluidity of the membrane is much reduced. Plasmid, those are bacterial cells, of course, that are suspended in a solution. Plasmid, once they've sort of been frozen, both physically and chemically, the plasmids are added to the solution, and then the solution is very quickly heated up. So the streams of warm material tend to flow from the warm to the cold, so from outside the bacterial cell to inside the bacterial cell, sweeping the plasmids along with them from the warmer solution outside into the cooler cell. The bacteria are then kind of left at their comfy temperature and comfy place to be able to recuperate, and a certain percentage of the bacteria that are exposed to this process are going to take up the plasmid or the DNA that we wish them to take up. So here's a diagram with a plasmid. We're showing plasmid and the bacteria inside the solution. We have, remember, the solution is calcium chloride, and we've got this is a very cool solution. We're going to heat them up rapidly, very rapidly, to 42 degrees, and hopefully when the warm liquid and plasmid solution sweeps into the bacterial cells, some of the plasmids are dumped into the bacterial cell, and then they go ahead and grow. Then what we'll do is we'll plate the bacteria on and in this case we have an antibiotic resistant gene kind of diagrammed in red. If the bacteria took up the plasmid, so that means they took it in and they're okay, they've got this great functioning plasmid inside. If we take those bacteria and we dump them onto a medium that has that antibiotic on it, they'll be able to live. So we'll know that we got the right, we got the bacteria that took up the plasmid, they're the ones that we want. If we put the bacteria onto this plate and they die, what we see then is they did not take up the plasmid, otherwise they would have been able to fight off the effects of the antibiotic, and they're not the ones that we want. So it's a good way for us to separate out the good, meaning the ones that took up the DNA we wanted versus the ones that did not.